doctor? Dr. Balakrishnan? Is Dr. Premalatha Balakrishnan with us? Dr. Premalatha?
Hello.
Hello.
Is my screen visible, sir?
Yes.
Yes, sir. Yes, sir.
Can you have the full screen?
Yeah, now I'm put. Yes, sir.
Okay.
Good afternoon, everyone. I'm Dr. Premalatha, working as a professor in the Department of Oral Pathology and Microbiology at Mahatma Gandhi Postgraduate Institute of Dental Sciences, Government of Puducherry Institution in India. I was, in fact, very happy to receive a mail from the conference secretary appreciating my article titled How Genetic Variation Was Analyzed in Phenytoin-Induced Gingival Enlargement Using Single Nucleotide Polymorphism of the Candidate Gene CYP2C9. So, which is published in the Indian Journal of Oral Maxillofacial Pathology, and it is published by the Indian Association of Oral Maxillofacial Pathologists. So at the outset, I thank the editorial team of the journal for publishing my article, and also the organizing team of the second international conference for permitting me to share my idea in this forum. And also I thank my head of the institute. My topic of my presentation is single nucleotide polymorphism to identify genetic variation. I'll be sharing my knowledge that how I have analyzed CYP2C9 gene in phenytoin-induced gingival enlargement by PCR RFLP procedure, that is polymerase chain reaction followed by the restriction fragment length polymorphism and multi-drug resistant gene in amylodipine-induced gingival enlargement by allele-specific PCR. DNA sequence of all human beings is 99.5% identical. Our DNA differs only by 0.5%. Does it make a difference? Definitely it makes a difference, because 0.5 difference translate into 15 million separate spelling differences in a genome of 3.2 billion bases. Genetic variation is the term preferred by the geneticists these days to describe mutation. And if the genetic variation is more than 1% in a population, it will be called as a polymorphism. Mutation in Latin means I change. In biological sense, it is a change in the nucleotide sequence as a result of the failure of the system to revert back the change, because there is a failure in the DNA repair mechanism. When you have a normal gene, it will result in a normal protein. When you have a mutated gene, it will either produce an abnormal protein or no protein. So genetic variation can be studied by two different ways. One is by using single nucleotide polymorphism. The other one is by using the copy number variant. 90% of all human DNA polymorphism are single nucleotide polymorphism. When you have a single base pair change, either by insertion or deletion, insertion means addition of one base pair to the fragment. Deletion means removal of one base pair from the fragment. Substitution means one base pair being substituted by the other. In case of insertion or deletion, it will result in a frameshift mutation. In case of substitution, it will result in a missense, nonsense, and silent mutation. You all know the human DNA consists mainly of 4 base pairs. 3 base pairs combine together to form one codon, which will synthesize one amino acid. 4 to the power of 3 gives us 64 different combinations. Leaving the start and the stop codon, only 20 different amino acids are produced. That means different codon can produce the same amino acid. When original DNA consists UUA, it will synthesize the amino acid leucine. In case U is been replaced by C, again the same amino acid leucine is produced, resulting in silent mutation. In case if U is been replaced by G, the different amino acid valine is produced, which results in a missense mutation. When a second base pair is replaced by A, then UAA will act as a chain termination codon. That means the further synthesis of the amino acid is stopped, resulting in a nonsense mutation. Example for a missense mutation is a sickle cell anemia. In the 6th position, actually it has to produce glutamic acid. Instead of that, valine is produced, resulting in the change in the morphology of the RBCs. Even if an allele of sickle cell disease is produced, it produces a clinical variation in a different population in a different way. That is, in Caucasian population, it produces a debilitating blood disorder, whereas it confers resistance to blood-borne parasitic infection in African population. For CYP2C9 gene, the variant ID which we wanted to analyze was this, and it is a single nucleotide polymorphism. It is related to the drug metabolism, missense variant, where 144th amino acid, it has to be arginine. Instead of that, cysteine is secreted in our case of CYP2C9. So the use of identifying genetic variation is to judge the genetic predisposition, to understand the pathogenesis, to evolve gene therapy in future or personalized medicine. For that, you have to know the difference between the genomic DNA and the target DNA. Genomic DNA is a chromosomal DNA of an organism representing the bulk of the genetic material, whereas target DNA is the specific gene in which we are interested is the target area. Importantly, the restriction enzyme do not cut randomly, rather they cut at the specific DNA target. The restriction fragment and length polymorphism are codominant. This principle has been utilized in the restriction fragment length polymorphism. When you see the genomic DNA, it is a coiled structure resembling like a muruku, the entire one. Whereas when you amplify the polymorphic region of the DNA using the universal primer, it will give the multiple fragment amplification. Whereas when you use a specific primer, only the specific fragment of the DNA will be amplified. The candidate genes are the sequenced genes of the known biological action, where you know the specific fragment. Gene will have the regulatory region and coding region. Coding region is one which codes for the protein, and the regulatory region controls, that decides whether the coding has to start or not. That is, on-off switch is present in the regulatory region. It has enhancer, upstream promoter elements and promoter elements in the regulatory region. Usually the regulatory region base pairs are represented in minus sign, and coding region will be represented in positive sign. Double-stranded DNA, by the enzymatic action, it becomes a single-stranded DNA. After that, using the law of complementary base pairing, the pre-mRNA is formed. Pre-mRNA will have exons and interons. Interons will be spliced within. Only the exons will fuse together to form a mature mRNA, which will be transported to the cytoplasm. In the ribosome, they synthesize the amino acid. So here, the synthesis of the amino acid AUG will be the start codon. one amino acid is synthesized. After that, every 3 base pair will code for one amino acid. Chain elongation takes place. When it encounters the chain termination codon, it will stop. CYP2C9 polymorphism, what we have analyzed is in the coding region. That is the reason here it is a plus sign, 430th base pair. The normal base pair for that position will be C. The polymorphic variant of that position will be the T. In 430 divided by 3 will give 143 plus 1. So that means the 144th amino acid which is affected. If it has a normal base pair, it will secrete arginine. In case if it is having the polymorphic variant, it will secrete cysteine. So it is a missense mutation what we have identified in CYP2C9 polymorphism. For MDR1 gene, 3435th amino acid, base pair, coding region, again C and T. T will be the polymorphic variant. Even if the polymorphic variant is present also, it produces the same amino acid isoleucine. That means it is a silent mutation for that position. We had one case of phenytoin-induced gingival enlargement and 3 cases of amlodipine-induced gingival enlargement. This was analyzed by CYP2C9 by PCR-RFLP and amlodipine by the MDR1 gene by the allele-specific PCR. This was a case based on the history, the gingival enlargement associated with the phenytoin intake. For that, the candidate gene, 2 types of candidate gene, either the biological candidate gene or the positional candidate gene. Positional candidate gene, where the linkage has been already established to the chromosomal position. For our CYP2C9, the established chromosomal position is the Q arm of the 10th chromosome. So CYP2C9, which encodes for the protein cytochrome P450, it is act as a metabolic enzyme. And for substrates like warfarin, tolbutamide, phenytoin, ibuprofen, and diclofenac, and losartan. CYP2C9 gene, which encodes for cytochrome P450, is a metabolic enzyme for the xenobiotic substances and the drugs. When you have a mutation or SNP in this position, it will have a reduced metabolic activity. So there will be a poor metabolizer resulting in an adverse drug reaction. This can be analyzed by the single nucleotide polymorphism by the procedure of polymerase chain reaction followed by restriction fragment length polymorphism. There are multiple ways we can genotype. That is depending on whether you know the single nucleotide polymorphism or you have to identify a new one. Known SNPs also can be identified by either by the gel-based analysis or non-gel-based analysis. We have analyzed the gel-based analysis, which is actually time-consuming and a cumbersome one. But nowadays, the recent COVID period and all also non-gel based analysis, that is the real-time PCR was used. When you don't know the SNP, you can go for a direct DNA sequencing. So polymerase chain reaction in 3 stages, pre-PCR, where mainly the primer designing has to be done and the Panstonix preparation. PCR is the thermocycling, then post-PCR analysis of the amplified product and the RFLP procedure. So for which the primer designing using the web resources with the help of the bioinformatics individuals, we have designed the primer and these are the master mix component. After that, it has been put into the thermocycler. Gel electrophoresis was analyzed. 690 base pair was the PCR product for CYP2C9. And again, the web resources chosen restriction enzyme is AWA2. Many enzymes are there. We have chosen AWA2 for this fragment. After the addition of AWA2, the gel electrophoresis was done. So web resources using the Ensemble genome browser, Primer 3 design software, sequence manipulation suite, and NetCutter mainly to choose the restriction enzymes. Restriction enzyme is nothing. It acts as a molecular scissors. It is a protein which recognizes the specific palindromic sequences. That is, the palindromic sequence recognized by the AWA2 restriction enzyme is GGACC. Palindromic means if you reverse scribe also, it will give the same meaning. So the PCR product in our case, 690, and the restriction enzyme which recognizes the site GGACC is in 521 position. So when it cuts, it produces the product of 521 base pair and 169 base pair. So this is the single nucleotide polymorphism site at 430th base pair, where you have to analyze whether C is present or T is present in that position, for which the primer designing, forward primer of 24 base pair and the reverse primer of 21 base pair was done. An allele is a variant form of a gene. An individual inherits 2 alleles for each gene, one from each parent. Wild type allele means it is a normal allele. Mutant allele means it is an abnormal one. Here, the site 2C9 has 10 exons, out of which exon 3, 430th base pair. The normal base pair for that position will be cytosine. It can be considered as a wild type base pair, which will be the star1 presentation. And in case if a variant base pair is present in that position, it will be called as a mutated sequence for that position, which is star2 presentation. Here, if a wild type is present, it will secrete arginine in 144th position, having a normal enzymatic activity. In case if it is replaced by T, it will result in cysteine, which will be having a reduced enzymatic activity. So isolation of the DNA was done from the 5 ml of blood, which is collected by disrupting the cell membrane protein denaturation and separation of the DNA from the denatured protein. And it has been recovery of the DNA has been done. So amplified PCR product plus the restriction enzyme AVA2 chosen from the website. It has been incubated at 37 degrees centigrade for 16 hours. Then polyacrylamide gel electrophoresis has to be done, and genotype can be identified based on the band size. In case if both the paternal and the maternal allele, they have the restriction site GGAC, then it will be recognized by the restriction enzyme and cut into 2 fragments, 521 base pair and 169 base pair. That means they have the normal base pair for that position. It is wild type. In case if both are having the polymorphic variant of the base pair T in that position, it will not be recognized by the restriction enzyme. So the entire fragment will remain intact. That will be called as an homozygous individual. In case if the paternal allele having the normal base pair, it will be recognized by the restriction enzyme. 2 fragments will be produced. If a maternal allele has the variant base pair for that position, it will not be recognized by the restriction enzyme. So 3 different bands are produced in case of an heterozygous individuals. So PCR product without digestion is given as a control. After that, wild type 2 bands are produced. And in case of a homozygous individual, single band. And in case of a heterozygous individual, 3 bands are produced. In our case, the first lane is the PCR without a restriction enzyme. This is after the addition of restriction enzyme. That means the restriction enzyme has not identified the site, resulting in a homomutant. So to summarize, 5 ml of venous blood, DNA extraction from leukocyte, polymerase chain reaction, check for the amplification and the digestion with the restriction endonucleases AVA2, incubated for 16 hours, then page electrophoresis was done. So it was a homomutant TT variant, that is both a star2 presentation. After that, we have done a genetic association study by measuring the plasma level of phenytoin. Actually, the normal level of phenytoin required is 15 micrograms. In our case, it was 33.5 micrograms. So after that, we reduced from 300 to 150 grams. And the gingival enlargement regressed. This gives an information he will be a poor metabolizer for phenytoin, but also he might be poor metabolizer for the other listed out drugs. Other gene which we have analyzed is the MDR1 gene, which codes for the protein P glycoprotein. It is a metabolic enzyme for amyloidopin. Here we have used the allele-specific PCR, in which case the forward primer will be common for both, and the reverse primer will differ for each fragment. So depending on the specific reverse primer, we will identify the type. When only the C allele is positive, that means it is a wild type. When T allele is alone positive, that means homomutant. Heteromutant means both C and T allele will be positive. The single nucleotide polymorphism analyzed position is 3435th. So we had HT, CT, both alleles were positive. It is a heteromutant case. In all 3 cases, we identified it as a heteromutant cases. So the difference between the PCR RFLP and the allele-specific PCR, primer designing In case of PCR RFLP, common forward and reverse primer for both alleles. In case of allele specific PCR, common forward primer for both alleles, but the reverse primer will differ for each allele. The interpretation is based on the bands produced, interpretation depending on the allele specificity. So we have used the web resources, software tools, and bioinformatics approaches to analyze and characterize the drug response mutation. This was one of the cartoon which I saw in the online. They said, my lord, they have discovered the human genome. So actually the hackers, we have to change the password now. According to me, one of the password may be the single nucleotide polymorphism. So after the Human Genome Project, basic tools exist to characterize the biology behind the human disease. Right drug to the right gene will help in the personalized medicine. Like how we are having a credit cards now, in future we might be having a genomic cards. This is the core team which has helped in my presentation. I am a part of the universe. I thank the universal energy for showering light on me. Thank you, all the participants, for your patience listening. Thank you.
Thank you very much.
Yes.
Dr. Premalatha.
Yes, sir.
Now, if there is any question,
yes, sir.
We are listening to you.
Yes, sir.
Does anyone have a question to ask Dr. Premalatha? It seems you do not have a question, doctor. So we
Yes,
thank
sir.
you.
Yes, sir. Yes, sir. Thank you.